as always, I would like to highly recommend to all of you to download apps and subscribe to my profile. Download the app as well on your phone so that you can receive all updates on upcoming episodes of this program if you enjoy it. All right, and now without further ado, let's go to the interview proper, because today it will be quite a little bit of a departure from our usual format, because as you can see, I am not alone here in my usual let's say studio, so to say, anymore, because I am joined by today's guest, medicinal chemist and professor of chemistry at the University of Milan, Professor Pierfausto Seneci, who also happens, very conveniently, I must say, to be my father. So, hi, Dad. Thank you so much for being here today.
My pleasure.
All right, so yeah, this is definitely. I've been looking forward to this one, I must confess. Like, it's not only not only for the family relationship, but also because I actually like to have like my guest in front of me in real life. So I start like jumping on planes every week to go from up to all corners of the world, which I would really love to do. Or I could bring all kinds of like crazy animal people in here day in and day out, which I'm sure you would enjoy too.
Well, up to a point.
All right, so the reason I brought you here today is because basically it's rooted in your expertise, because you to give a little bit of background, like you graduated in chemistry from the University of Pavia in 1983.
A long time ago. Let's say a long time ago. That's
exactly. And then since then, you have worked in quite a few different institutes, and right now you have been as of now you have been teaching at the University of Milan for around 15 years.
Yes,
something like that. And you, of course, you have been you've been very active in the drug design and drug discovery field in the last few decades. So I remember one of the things that you would always tell me when I was little, and my passion for snakes, reptiles, and all kinds of scaly critters was coming up. One of the things that you stressed to you used to basically like bond with me in that sense, to like find a common ground, was the fact that many molecules and compounds that actually originate from these animals are used, or at least have been explored for any medicinal potential in pharmaceutical research, especially in the last 50 years. And this is definitely something that stuck with me so much that now I am basically invested in that field as well, although more on the animal side of things than the pharmaceutical side. So I was thinking, first of all, just to start, because when we think of toxins, of poisons and venoms, of course, we have a picture of compounds and substances that are evolutionarily designed to injure or kill either prey items or predators or aggressors. But we wouldn't be immediately inclined to associate them with drugs and medicine. But actually, the line between the two is a lot thinner than we might think. So why are venoms and toxic compounds so interesting from a medical point of view?
Well, let me take this a little bit more from the beginning. We are talking about natural compounds. That's one of the areas in which I've been more active. So compounds which are biosynthesized by living organisms, rather than synthetic compounds that can be synthesized by the synthetic chemist, organic chemist. Obviously, those natural compounds, they, as you said correctly, have been developed by evolution, which is, we always joke among chemists, the best rational drug designer, simply because they are made for a purpose. As you said correctly, the purpose may be to kill the prey, and that is where the snakes and other animals play in, but it could also be to defend themselves for animals that do not have any other kind of weapon, and that's the example, another of the example that I'm sure you remember from when you were a child, the example of Epipedobates tricolor and frogs and other amphibians. But from a more general perspective, there's a need for any type of living organism to have a competitive advantage in its environment. And if we talk about natural compounds, that's what they provide. And some of those, not all of those purposes, but some of those can also be exploited by humans and can lead to new drugs. The typical example, and I apologize if it appears to be boring, has to do with a guy, you probably know his name well, and his name is Sir Alexander Fleming. 80 years ago, roughly, he published on a relatively obscure journal a paper related to an entity, a chemical entity, which was called penicillin. And at that time, Sir Fleming was convinced that this was a protein and that could have been useful to become a topical antibacterial to cure infections. One thing that needs to be said at the beginning is antibacterials as a concept did not exist at that time. So if you got an infection, serious or mild, you could die. It depended only on your immune system to be saved or not. While all the story of penicillin led Fleming, other people really, but that's a long story, to discover the first antibacterial and to start curing people. That's because the producer, which is Penicillium notatum, which is a mold, has a competitive advantage being able to kill the other microorganisms in its microenvironment. So it's an advantage, competitive advantage developed by nature, by evolution. But we can take advantage of that for two major goals coming from any type of natural product producers. One of them is to kill bugs, so infections, and the other one is to kill proliferating cells, and that's mostly oncology. If you take a look today at the portfolio of drugs that we have available to cure tumors and or to cure infections or also viral infections, more than 50%, I'm saying more than 50%, more than 1 out of 2, they are either natural compounds as such or synthetic modifications of natural compounds, which means without this source of compounds, innovation, which cannot be really predicted because nature is very complicated in developing structures. We are good in rationally designing drugs now, but not as good as nature. We will never be, probably. We have a lot of tools, we have a lot of possibilities, but natural compounds are very valuable for drug discovery. That's a fact.
Oh yeah, absolutely. And you need
let's talk about venoms. You were asking me about venoms, right? Well, you know, another thing that I would like to introduce here, and I hope I'm not behaving as the typical professor. That's supposed to be.
Oh no,
that's supposed
me,
to be. That's supposed to be.
Definitely not.
That's supposed to be a sort of a discussion. The border between drug and poison is very fuzzy. It's really very fuzzy. There is no absolute drug. Any drug that is taken at different dosages, at wrong dosages, can be a poison. Vice versa, any compound that has toxic effects in an appropriate formulation, in an appropriate dosage, in an appropriate situation, then can have positive and curative effects. And that's exactly the example of, for example, components of some of the venoms, about which you know a lot more than me. So I will not.
No, yeah, definitely. Like that goes all the way back to Paracelsus in the 15th century, who first, well, his like most famous quote is, of course, he wrote it in Latin, but now it was basically something like it is the dose that makes the poison, because even basically any drug that we even any over-the-counter drug can become a poison if like you take insanely high doses of it, or if you mix it with the wrong substances. And so basically, yeah, that's what I was talking about indeed when I said that the line between toxins and medicines is a lot thinner than many of us think. And indeed, so well, you mentioned penicillin, which arguably I am like very like more multicellular organism oriented, so I didn't immediately think of it. But
if I may comment upon that, one of the things that natural product related researchers have to think about is a process. There's a process to get to a natural compound and develop it as a drug. And out of this process, there are steps which are much simpler if the producer is a simple organism, what we call lower producers, not in terms of how much it produces. Actually, it produces more, but it's lower in terms of evolution. So we are talking about bugs, we are talking about bacteria, monocellular, or we are talking about lower eukaryotes, molds, mushrooms, and those kind of things. Why is that? The point is, those compounds often have very complicated structures. I don't know if any of you has ever heard of some very popular drugs available today, which are the taxanes that come from Taxol, which is a natural compound, which is a nightmare. Yeah. Chemically speaking, that compound is a nightmare. You can take any Nobel Prize in chemistry or any other discipline, which claim that they're very good, and they are, because they got the Nobel Prize, but they could never have been able to rationally design Taxol to bind to the binding site to which Taxol binds in order to exert its activity. So that's what nature is. Having said that, if you're working with a microorganism, as we are as human beings since thousands of years, you think about bread, you think about beer and wine and fermentation, that's how you grow cultures of microorganisms. Now with molecular biology, you can play with their genome and increase their productivity in a biologically active compound, and you can make kilos, even tons, of antibacterials, anti-cancer compounds, or anti-malarial. You may have heard of artemisinin, those kind of compounds. And it is very cheap. Vice versa, Epipedobates tricolor, our old friend when you were a kid and I was a young,
just a little bit of context. Epipedobates tricolor is the scientific name of the phantasmal poison frog, which is a tiny little amphibian that lives in the rainforests of I think Central America. I'm not sure if South America too. But yeah, exactly. And honestly, I don't even know why they called it tricolor, because it only has two colors. It's like red and white. So whatever. But still, it's a highly poisonous frog that, like most, if not all, dart frog gets its poison actually sequesters its poison from the insects it eats. So now back to one of the...
there are 4 steps that you need to undertake, and they are very complicated, to discover a new drug coming from natural sources. The first one is to identify the producer. So, as you probably, many of you that are hearing here, I don't know how many, but many of you have some biological knowledge, and you know that it's enough to have a petri dish and a culture of a microorganism, and you can check a lot of things about this microorganism. For example, if it kills another bug. So it's simply a very simple in vitro test. Vice versa, if you are screening higher organism, being flora or fauna, the one you prefer, you need to see an observation. There needs to be an observation, a macro observation, like Epipedobates tricolor is typically the example I use. The skin of these frogs, which is secreting some white powder, which is used by Indios in the Central America area to dip their the tips of their arrows and to kill people. So those type of observations are much more difficult. But that's not the major issue. At the end of the day, when you need to produce kilos and then even more than that to produce Taxol or artemisinin and so on, you need to be able to really increase the productivity of the producer. What about a bug? You can change the genome, suppress all the other mechanisms so that the E. coli or the likes of the E. coli, they die while they produce it. So what? I mean, cultures of E. coli, cultures of microorganisms, there's no moral issue on that dying. And you can make fermenters and make huge amounts of erythromycin, penicillin, and so on. Vice versa. Think about the skin of frogs. Think about the bark of a Taxus brevifolia, which is a tree which produces Taxol, which is now, taxanes, which are derivatives of Taxol. It is one of the best drugs to cure tumors. They made a calculation at the beginning in the 90s when Taxol entered the market. In order to be able to have enough Taxol from natural sources for clinical experimentation, I'm not talking to cure people, just to check that it works, they would have deforested all North America so that you would have had extinction of Taxus brevifolia. And then? So you see, the issue is not only to find the producer, is not only to find the good molecule, but also to have enough of them, either from natural sources, typically microorganisms, or by putting together some complex processes that allow you to arrive to those molecules. So there's a lot of work. Nature is fantastic, as we said, but there are things that people like me can do and nature cannot, believe it or not. It's like that. Nature has a major constraint. It has a lot of advantages, which is being able to be very specific in biosynthesizing complex molecules. You know about enzymes, those kind of things, which are exquisitely selective. But the issue is nature is confined to chemical reactions that take place in water at neutral pH. Why is that? Because otherwise the organism would die. While we, under the hood, we can use any solvent, any temperature. So if you put together the innovation that comes from nature, the compounds, the new mechanisms like penicillin, but then the capability that we have now to modify molecules and to make them more, well, for example, less toxic, more bioavailable, more specific, and so on, then it's sort of a perfect marriage. That's why natural products and natural compounds are useful, but having the possibility to play with them and to characterize them is also very important.
No, yeah, and this one thing that I was wondering about is that you mentioned, the case for the poison frogs. I don't know if the Epipedopathes tricolor specifically, but definitely a few species of those were long known by Native American populations as for their toxicity, and indeed they used to coat the points, the tips of their arrows in the poison to hunt, to take down monkeys and other prey in the rainforest. And again, what I'm thinking is that some medicines also come from compounds found in snake venom, like captopril, which is probably for decades it was the main antihypertensive drug on the market, and that comes from a protein isolated from the venom of the Brazilian lancehead, a pit viper from a South American pit viper. And you were talking about observation, so they need to actually recognize that that's something, that the effects of these molecules on a living organism. And sometimes these effects can be sometimes this observation literally consists of realizing that these toxins can be deadly to humans as well. Because absolutely, of course, we all know that snake bite is a bite from a Botops jararaca, from a Brazilian lancehead, could definitely, if not kill you, at least send you to the ICU right away, because oh boy, that can definitely reduce your bloodstream, your coagulation system in shambles. And indeed, that's basically how someone at Instituto Butantan got the idea of testing whether some of these compounds in isolation, again, in right conditions, can actually work to lower blood pressure.
That's exactly my point, Lorenzo. The point of, I always stick to my examples that I use teaching, but coming back to Fleming, that has claimed a lot of things which really should not be attributed to him. But what is incredibly
that also happens a lot in science.
Incredibly clever of, you know the story probably, incredibly clever of Fleming was that coming back from holidays, he found a lot of dirty petri dishes, and rather than throwing everything away and washing it, he noticed that there were dishes that should have contained some pathogenic Staphylococcus aureus, and the aureus was dead there, and there was another mold. And his cleverness was, let's understand why this happens, rather than washing everything. It was not something that it was expected. That is also why the attitude of people looking from natural compounds to find something with usages in medicinal chemistry and pharmaceutical research or somewhere else is to really keep your eyes open and be prepared to the unpredictable. At that time, it was unpredictable. No one believed that it would have been possible to eradicate infections by using antibacterials because they did not exist. Some other examples of natural compounds with this type of effects, also, as you said correctly, by really taking a closer look to things that normally were not considered as a potential source of drugs.
No, yeah, indeed, and again, it goes back to it also goes back to what you said before about the fact that some molecules that we find routinely in nature are something that no one in their right mind would ever think of constructing in a laboratory setting, like Taxol, which is an absolutely biochemical nightmare, and also I could think about Suitarin C, which is an an enzyme, serine protease found in the venom of some Australian snakes, and that is basically mimics the complex composed of two coagulation factors that we also have in our blood. And it is absolutely insane that actually this was replicated in the venom gland of a snake to act as a toxin. That sometimes these molecules are absolutely mind-blowing in their complexity, in their structure, in their size, and such. So
one thing that I would like to mention here, another relevant point, going back to Epipedobates for a second, which I love, probably you have noticed.
Oh yes,
is that sometimes
should have brought a picture of that frog.
Sometimes, well, you may show it afterwards. Sometimes it's not like you're really looking for a drug or you're finding a drug, but sometimes you find something which is as valuable, which is you really understand how a toxin works, something which is toxic works, and you elucidate a new mechanism connected to a disease or connecting to something that can lead you to acquiring information that will eventually be used with different molecules to provide benefits. And the typical example, again, is Epipedobates. This guy produces this white powder. Typically, amphibians produce either peptides or alkaloids. Here we are talking about an alkaloid. And this alkaloid is called epibatidin. That's why Epipedobates, that's why epibatidin is called epibatidin, because of Epipedobates. And that molecule cannot be a drug because it's extremely potent and toxic. But this molecule, which is very simple, by the way, once you have elucidated the structure, then you can synthesize it, so you don't need the frogs and their skin anymore. That molecule is 200 times more potent than morphine as a nociceptive agent. So it is so potent because it binds to a specific receptor, which is an acetylcholine receptor, and elucidating the binding of this molecule to that receptor has shown to people one way to tackle some diseases via symptomatic treatments for Alzheimer's. So is this molecule a drug? No. But was it useful? A lot, because we understood a lot of things.
No, yeah, I also have. I was about to bring that up as well, because indeed, sometimes even aside from the purely like pharmaceutical aspect, where it's about making a drug out of a toxin. Sometimes, toxic compounds have also served an invaluable function for diagnostic purposes, and also for elucidating like how certain mechanisms of our own physiology work. Like think of like when in the early 60s, a molecule now known as alpha bungarotoxin was isolated from the venom of the many-banded crate, extremely highly venomous snake from Southeast Asia. That was an alpha neurotoxin, which, to cut a long story short, blocks neural transmission from synapses to muscle fibers, and it so induces paralysis because the muscle no longer receives input from the nervous system and therefore cannot move. And that has been used indeed. That toxin is maybe not the only reason, but one of the main reasons why we know this in the first place, because it was shown that this toxin acts on specific receptors on the muscle fibers, blocking the influx of sodium ions and depolarization. Yeah, it's a long story, but still, it really helped us bring light on how our own body operates. Let's
put it this way. I understand. Rationally, I understand. One thing I should say as a disclaimer at the beginning, I'm scared by snakes and things. So you see that, evolution and heritage of traits also diverges a little bit. So I'm a little bit different from my son, which is good and bad in a few things. But having said that, I really believe that there are a lot of reasons why biodiversity and chemical diversity, because you're very much, guys, interested in biodiversity, right? In some specific areas. I'm very much interested in chemical diversity, and the two things are connected. The fact to have on our planet as many as possible living species can also be exploited. And when I say exploited, I don't mean it in a negative term. Can be exploited without damaging the different species to help the quality of life of people. In my case, I'm talking about drug discovery. I'm talking about finding new active principle to cure some diseases. But you could also think about this type of natural compounds or construct or produced by any organism in other areas. We were talking with Lorenzo in the past about silk and about things like spiders, the spider's web. Those kind of proteins have given a lot of ideas about to researchers, to be able to create some synthetic materials that resemble that. So there's a lot of opportunities coming from natural compounds. And one thing I would like to underline is, many people may believe that we are scientists. We believe that we can do anything. We have a lot of rational tools. But one thing that should be considered heavily, and I believe that it is luckily considered heavily, is traditional medicine, for example. So exactly what people do since thousands of years, simply by observing what happens in nature, by observing other animals, what they do, by understanding, for example, that the roots of the willow tree could be useful if you make some concoction with that to reduce inflammation. Why is that? Because aspirin is inside that. So a lot of traditional medicine has foundation on compounds which have very clear and powerful biological activities that could be exploited by becoming drugs, as we have done in the past. So there's a lot of knowledge around coming from tribes and populations that still needs to be.
No, absolutely, and and I also think this is a good response to the this new wave of people like obsessed with like so-called natural medicine, who basically refuse to take any, basically distrust any like classical big pharma thing because it's not natural. And I'm like, dude, like you said, it's over 50%. You said over 50%.
In some areas, it's over 50%.
Yeah, absolutely. Exactly. In certain areas, over 50% of like the even over-the-counter drugs are obtained literally from natural sources. So the whole non-natural argument already dies right there. And then, of course, this doesn't mean that you can cure cancer with plant roots or something like that. But still, you're totally right that some venues offered by traditional medicine and traditional ecological knowledge.
I would make a comparison. Definitely. I am sure that I don't want to know it for personal and emotional reasons. I don't want to know what you do when you go out looking for black mambas or things like that, because I would be scared. I couldn't do it. But when you do that,
story, but like,
when you do
that's for later, perhaps. Yes.
When you do it, obviously you are very interested in finding, observing, and looking at. But you're also always very careful. Am I correct? Because you know which is the danger connected to that. So it's the same story. I'm here to tell you that nature and natural compounds are extremely important. But if you tell me that anything which is bio and natural is fantastic and you should take it without thinking about it, that's a big mistake. We're talking about toxins and venoms. The best and most powerful venoms are not synthetically made. They're natural, and they are in natural environments. Even an organism that produces something useful, if you change its microenvironment, and if you change its food, typically something different happens. And there are toxins that in stress conditions are produced by some plants, for example. So those kind of things can be very dangerous. Even eating only biomaterials can be very dangerous, because there are natural compounds which are extremely toxic in some conditions on some people. So it is always a good source of things, but you always need to take care of what you're doing, why you're doing, how you are, for example, identifying and extracting an active molecule, how you purifying it before eventually, possibly reaching the market as a new drug.
And yeah, another thing that I was going to ask you is again back to the like hugely complex molecules, which again I now realize I might have given a little bit of a distorted perspective, because while it is true that some of these molecules are mind-blowingly complicated, many, if not most of them, are remarkably simple on the other hand, like very small, very like simple structure and everything. And that also serves an evolutionary purpose, because they are easy to produce. They don't require a lot of energy to invest in their production. So that also benefits the organism in the first place. So that also must be taken into account. But then again, sometimes
let me give you an example on that. We were talking about Taxol. If any of you wants, you can go and take a look at the molecular structure of Taxol, which, when I say it's a nightmare for a chemist, it's not because it's particularly large. I mean, it's much smaller than protein, for example, any simple protein. but it is really complicated in terms of connections between atoms, stereochemistries, chiral centers, hindrance. So it is something that, chemically speaking, is very difficult to assemble from symptoms, from starting materials. While Taxus brevifolia can make Taxol via its biosynthesis, its metabolism, and with the current production, we can make from natural sources, playing a little bit with that, we can make tons of Taxol. We have currently 5 or 6 total syntheses from simple material to Taxol, which have been developed by the best minds in organic chemistry and medicinal chemistry. 1 gram of Taxol made synthetically could cost in the range between $10 and $100 million, while 1 gram of Taxol biosynthetically made is in the range of a few dollars. So you see the difference. You see the difference. When the molecule is complicated and it has been evolutionarily optimized by nature, if you can produce it naturally by playing with the lower organisms typically, then you can make very large amounts of penicillin, erythromycin, you name it, cephalosporins, any of these antibacterials or Taxol or other things, with a very cost-effective process, which will allow, and that's important, will not only allow big pharmas to make a lot of money, which is also true, but it also will allow millions of people to be able to afford these molecules. The social systems and the health systems, not only in the US of A, but also in other countries, will be able to afford curing people with those drugs.
Absolutely, but then another thing that I'm thinking about is the flip side, so to say, the other side of this matter, which is, of course, we said that many compounds are sourced from venoms and toxins have become incredibly successful drugs, but it's not all rainbows and butterflies, because many more have been explored for their medicinal potential, but they couldn't make the cut for the market for various reasons. And indeed, one thing that I wanted to ask you is, what are the main constraints, the main limits that a researcher might encounter when testing whether some natural compound can become a drug? So what could basically screw everything up in that sense?
There are some classical ones, which are also the classical obstacles and hurdles that are encountered when you try to develop a new drug. Often, not always, but often, it is not to do with the molecular mechanism of action. It's not because they are not acting on a significant target to cure a disease. It is more related to the fact that they need to be, what we say, bioavailable. So you need to be able to administer them to the patient and to have them reaching the binding site, which may be into cells, organs, and so on, in an appropriate manner with no toxic effects. So often, natural products are produced by fungi, mushroom, or anything for internal purposes of the organism, then they can be excreted. So they are not really optimized to be bioavailable. They don't need to be bioavailable to be very good toxins and venoms. So often, what we need to do from the natural compounds is to change them a little bit. What do I mean with a little bit? Not enough to make them less potent on their target, which is a protein called tubulin for Taxol, for example, or some proteases for penicillin, but you change it a little bit so that it still binds there, it still has the curative effect, but it becomes more bioavailable, which means mostly being stable in biological fluids so that it enters the bloodstream and it's not metabolized by anything. It doesn't, it gets not metabolized by the liver and those things, but even more important, it becomes able to cross biological membranes. Let's take again the example of Taxol, which is this fantastic molecule, which has allowed us to do a lot of things. But Taxol per se is not at all soluble in water. It's very lipophilic. So can it become a drug? No. Not a drug that can be given to people without using IV infusion, which is not ideal, would be typical for hospitals. While some Taxol derivatives, which have been made by modifying the structure of Taxol, introducing some small modifications that increase water solubility without losing binding activity, those compounds are much more bioavailable, so they have become much better drugs than Taxol could have been. So are those molecules synthetics? Yes. Could you make them without discovering Taxol? No way. So you see the marriage of the two things. So typically, to come back to your question, it is more related to bioavailability. You need to, when you have a drug, it does not only need to be selective and specific for a target, a protein, which is the one that you need to modulate. It also needs to be bioavailable, non-toxic, cost affordable, because, as I said, $10 million for a gram, even if it would be a fantastic drug, even if there would be millions of people needing it, no one would be able to produce it, because where do you get the money to make this? Where do you get any social or health system able to afford it? So that's another of the major goals that we have. Find something which can be manufactured with proper specificity and accuracy in terms of characterizing purity and so on, but also with a cost affordable process. So those are the key points: bioavailability, specificity, and effect. It's a very long process and it's very expensive. That is something that people tend to neglect. It is a very expensive process going from an idea to a drug, because there's a lot of preclinical and clinical things you need to do. And it's becoming more and more expensive. Why is that? That's because you're more and more interested to avoid any side effects to people. So it's really very expensive. And for that reason, only entities which have a lot of money are able to afford those kind of things. So that the starting point, any source, natural compounds, or rational drug design need to be taken forward in a process which is very complicated. And often evolution and nature give you an advantageous starting point, which is still the case today. You may be surprised, but would you be able, guys, to guess where typically companies based on natural compounds are proliferating currently? Would you think about US?
Geographically? Well, yeah. Feel free to show us your to answer with your guesses in the comments. By the way, while I think about it myself as well too. So where geographically
where there's more biodiversity.
So basically in the tropical belt.
For example, in areas in which, especially as I said, microorganisms, but also flora and maybe fauna, where you have the most variety and the more different conditions, which are also typically the areas of the world where systematic search of natural compounds has never been made. Why? Because they are not wealthy areas of the world. They are areas of the world which are typically underdeveloped, and for that reason, there are people which have not really exploited those as potential sources of natural compounds. This would be, but probably we don't have the time and it's not relevant to you. This could also be used to underline the unfairness of many of these processes. Just to mention, there are some countries, very powerful countries, that have exploited areas of the world in which there's a lot of chemical diversity that can become drugs. They have been developed by the companies that found them there without any recognition, any financial recognition to the countries.
Absolutely.
Those kind of things now should be prevented, but it's still difficult.
No, indeed. Yeah, that's actually, thank you for bringing that up, because it's definitely a major ethical issue connected to to pharmaceutical research in general. Right. Even aside from the toxin field. Correct. And now something a bit more. This question is a bit more specific on what you on your personal work, and then we'll go back to the bigger picture in the end. So in recent years, you've been working closely on neurodegenerative diseases, which are perhaps one of the most pressing health challenges for global health,
especially when you get older.
Worldwide, in our times, because it's they are getting more and more widespread and appearing with an ever increasing frequency. So, is there, like, to your knowledge and even in your personal experience, are there any like interest, at least promising venues for research on treatments for neurodegeneration that might come from the biodiversity the biodiversity aspect of medical research.
If you remember what I said at the beginning, you always need to make a connection, and the connection is you will be able to find something if there's a reason why nature would have evolutionarily developed the biosynthesis of a compound for relevant interest of the producer. So again, the example of Epipedobates. Why is Epipedobates producing a neurotoxin, a very potent neurotoxin, which is epibatidin, on its skin as a passive defense mechanism? So typically from these type of organisms, like amphibians, but also snakes and scorpions and spiders, we have found some neuroprotecting agents. One of them is epibatidin, understanding the mechanism of action of inhibiting acetylcholine receptors and obtaining some effects. I'm sure that we also spoke about it several times. You're familiar, and some of the people that are listening here are familiar with mambalgins, for example, psalmotoxin, those kind of materials which are extremely toxic, those proteins that come from snakes, that come from scorpions, and they have been very useful to characterize their target, which is an ion channel, which is the acid-sensitive ion channel, or ASICs. So there are examples of these neuroactive molecules that can also be neuroprotective. The last and very final comment I would make is most of neurodegenerative diseases, Alzheimer, Parkinson, Huntington disease, amyotrophic lateral sclerosis, we are talking of horrendous diseases. You can have some symptomatic treatments which delay the disease. You would hope to find some disease modifiers that block or even regress the disease. In terms of symptomatics, there are several of those molecules which are known since a long time. All what you can do now, if you get Alzheimer and if you have Alzheimer, to lower down the speed of progression of Alzheimer, all of this has to do with natural compounds. Galantamine, for example, is one of them. They all come from plants, and they all come from specific metabolic pathways of plants, and they are useful to lower the disease. To block the disease, often you have proteins which have been aggregated, they have been misfolded, and so one of the areas in which we are working the most now is to look for compounds which are able to disaggregate molecular aggregates of amyloid, Alzheimer's disease, amyloid protein. So are there natural compounds developed for that? Found and then further developed? Yes, a few of them. None of them to be really found in the development cascades and pipelines of big pharmas that I know, but it's a good source. The final comment I would make is there are other natural compounds that we could consider in broad terms natural compounds that come from therapeutic avenues which were not available 30, 40 years ago. I'm talking about, for example, molecular antibodies. I'm talking about proteins which have been developed with the help of the advancements in biology and molecular biology from natural sources. That's an area which is very popular at the moment in therapeutic approaches towards neurodegenerative diseases, to make molecular antibodies which are able to go and bind to the amyloid protein, to synuclein in Parkinson, and so on. All those things have a natural component. Specifically about snakes, amphibians, and specifically about compounds coming from that, honestly, I'm not that aware, apart from the example I gave you for some ion channels. Definitely some ion channels have been characterized, and people are now able to rationally look for molecules modulating those ion channels because of the mambalgins, the psalmotoxins, the epibatidin that I mentioned before. But this is not to say that it's impossible. Nature is very clever. Nature is very varied, and there are a lot of organisms, and I'm sure that in future it will make sense to look for these kind of active principles also from these sources.
Oh yeah, and this ties nicely with what I was planning with my last question. And indeed, like just before that, I'd like to remind the audience that you can also ask questions here in the comments. You can just type it in, shoot us a question, and we will my dad will answer it to the best of his knowledge. So you said indeed what you just said, Thijs, is definitely reminds me of something that my former and hopefully soon to be again supervisor, Dr. Brian Fry, used to say a lot as an argument to promote conservation of venomous and poisonous creatures. So, which basically is, you never know where the next wonder drug could come from. So if even if some species that hasn't even been described yet goes extinct because you completely mowed down the forest habitat it lived in, and then that species indeed synthesize some compound that might have had huge pharmaceutical potential, then that is, and if that species goes extinct, that is lost forever.
That's a very good point, because as we said, traditional medicine, which is extremely relevant even today, exists since thousands of years. What is different is not the purpose of what traditional medicine did and what medicine does today. The purpose is always to provide benefits to patients. The difference is in technology, what we have available in order to look for active compounds. So that is why exactly what you said is, and what Brian says is very correct. There may be some producers of natural compounds, that may be some snakes, might be some amphibians, that we cannot even properly estimate today because we don't have the tools, the assays, and the ways of checking and understanding the fate and the effect of something that they produce. We don't have it today, but maybe we will have it in 10 years, in 20 years. And, as you said correctly, if we lose them, it's gone. Same thing as the Epipedobates tricolor or the Taxus brevifolia or any of those producers.
Absolutely. So, and this makes me wonder, because I can definitely imagine that a lot of people are not even aware that some of the drugs that they might be taking or they have taken actually come from sources of this kind, like again captopril for antihypertensive that comes from snake venom, or even that is recounting like my personal experience. Until last year, I had no idea that the drug called Byetta or Bietta, I don't know how to pronounce it, which is a common treatment for diabetes, comes from the venom of the Gila monster, which is like an extremely odd-looking lizard, so to say, that lives in the desert of the southern United States and Mexico. So yeah, exactly. I didn't know myself until not too long ago. So do you think there could be a way to, like, even speaking from like the more entrepreneurial or pharmaceutical side of things, to improve on this front, so to make to raise awareness of where actually these treatments potentially these life-saving treatments come from, so that to bridge the gap between the consumer and the source.
That's a good point. It should be people should be more aware of where their wonder drugs come from. That's for sure. I concur with you that it's not that well known, apart from very generic statements like, as we said before, there are people that believe that only bio is good and anything which is not bio is not good. That's an approximation which is not absolutely correct. It would be more important to educate the people, respecting bio and chemical diversity, to have this source of potential useful things, tools available for the rest of their lives, the lives of their son and grandsons, and so on and so forth. How to do this? Well, that's difficult.
Oh yeah,
that is very
Tell
difficult.
me about
that. because well, obviously it's not the right place and the right venue now to talk about it. But I'm sure that I'm not saying anything surprising to anyone here if I say that we live in a capitalistic society, which means that profit is needed, especially for private endeavors. You were talking to me before, talking about entrepreneurial activities. By definition, you need to set up something and you get the money to do something only if it is promising. For promising, it means not only to be efficacious, curing people and so on, but also promising in terms of making money and preserving the life of the company that can continue to make research and hire people and develop scientists and all of that. So the two things need to be connected. So there are some, to give you an example, we were talking about neurodegeneration. I believe and I hope that everyone would agree that neurodegeneration is one of the major problems of humanity in future, because more and more people get old, which is good, and more people get to experience, unfortunately, Alzheimer, Parkinson, and so on. And there are no effective drugs available. So it could be something of a surprise if I would mention to people like you that not that many companies are researching that. And it's not because it's not essential. It is essential. It's because it's very expensive and very dangerous in terms of it's difficult to find something active. This means you're wasting millions and millions of dollars without reaching at the end a drug, but just hitting the wall as all the other companies have done. So in areas like this, there should be something that should become a little bit less capitalistic, if I may say, promoted directly by public entities that do not have to write down their pros, their benefits, and entries and exits in a balance sheet. They can put money for research and stimulate people in different areas: chemistry, biology, molecular biology, natural sciences, looking for new principles and financing those endeavors, knowing that this money will not immediately become valuable in selling drugs, but opening up new avenues, understanding new molecular mechanisms to cure Alzheimer and so on, and making people aware of the fact that they need to preserve nature, they need to preserve biodiversity, and they need to think about how to proceed on those areas in a bit of a different area. Mindset, I would say, at least. I don't know if I really answered.
No, absolutely.
But that's the comment. We are not very attentive to those things. We are more attentive to how many different varieties of smartphones are available, or the type of car you want to buy, or the clothes you put on. I'm fine with that. But again, if this is the major goal of humanity, I think it's nothing that relates to me because I'm old, but I think we are not really moving in the right direction. That's my opinion.
No, but then, so basically, bottom line is don't forget to fund people to just go out there and look for some kind of go search for some kind of poisonous or venomous animal and see what the heck that thing does. So yeah, I have no vested interest in that, of course. Absolutely, no conflict of interest here. But yeah, so basically that was it, or at least a big part of it. So yeah, I see that there are no more questions from the audience. So we have reached the end of our interview, and once again, thank you so much for being here today.
It was a pleasure. It was an experience.
Oh yeah, absolutely. Yeah, for me too.
It wasn't
absolutely,
and boring for the people.
No, I enjoyed it a lot, and I found it extremely instructive. And yeah, again, I'm not saying this just because you're my dad. I really mean it. I wouldn't even need to. I wouldn't even need to.
There would be no reason to
exactly.
do that. You could only be disinherited. That's the maximum.
I mean, this must be the millionth time you've done that. Like, come on, you've lost any credibility in that sense.
People don't know that.
Well, now they do.
Correct.
So yeah, that's again, thank you. It was
my pleasure.
It was a great pleasure. And so for everyone else in the audience, you might have noticed that lately I haven't been as active as I would have liked to here on the Snakeman's Lair, because in all honesty, it's an extremely busy. These are extremely busy times for me. So it might not be as regular as usual. So not necessarily once a week for a month or so, but I will try my best to stick to the schedule, and I'm already in contact with potential new guests. So definitely stay tuned, and yeah, stay alert for new updates. Download the apps app again, and you will get all info from there. So yeah, I hope to see you all again for the next episode of the Snakeman's Lair. And in the meantime, I wish you a great weekend. Thank you again for being here, and goodbye.